So in this video we're going to think about how you do restriction cloning. So cloning is the process of moving bits of DNA around into combinations that you want, and restriction cloning is kind of the basic form of cloning. There are now quite a lot of alternatives to restriction cloning, but it's still used in labs all around the world, and this is the sort of fundamentals of cloning. So we're going to think through this process. So with cloning, what you fundamentally need is two things. So one is a plasmid. So that's a small circle of DNA that bacteria use to sort of shuffle genes around. There's a whole other video about plasmids that you might want to have a look at. And that plasmid is what we know as a vector. So a vector is something that can move something else around. So the mosquito is a vector for the malaria parasite. So the mosquito moves the malaria around. In the same way, the plasmid is going to move our piece of DNA around. Okay, so we've got our plasmid vector. Plus, we've got our gene of interest, which I'll just abbreviate to GOI. Okay, so this is the gene that we want to amplify, or we want to get from one species into our plasmid vector. Okay, so we want to combine these two things together. Okay, so the first thing that we do is to digest with restriction enzymes. Okay, so what this means, and there's a whole other video on restriction enzymes. Basically, we cut open that plasmid vector, so it gets cut. So there's a restriction site on the plasmid vector. We cut that open, and we digest our gene of interest with the same restriction enzymes. And I'll talk about how we do that in a minute. Okay. We digest them with the restriction enzymes, and then we have to purify them. And then what we do is we ligate them together. So we basically stick them back together. So I've now got my plasmid that looks like this. So let's say that this plasmid was 4 kilobases to start, and my gene of interest is 2 kilobases. Okay, I've now got a plasmid that's been cut open. We've popped the gene of interest, and we've now got 6 kilobase plasmid into is my vector. So I've got the plasmid background and I've got the gene of interest. I then transform into cells. So that's usually E. coli. So I've now put so here's my E. coli cell. So the E. coli cell will have its own chromosome, where it has all of its normal DNA, but we can add this extra little bit of plasmid, so we can put that into the plasmid. So put the plasmid into the cells, and the bacteria will start to copy it. So we've got multiple copies of that plasmid. And then we can extract the plasmid and verify it. So we might sequence it, for example, to check that it's right. Okay, so that's the process. Okay, so we've got our plasmid, we've got our gene of interest, and that is amplified by PCR. So the polymerase chain reaction we use to make a copy of the gene of interest. That gene of interest, we digest it, we stick the two together and we ligate it. Then we've got our plasmid, we transform it into cells, and then we can extract and verify the plasmid again. Okay, so that's the basic process. So let's think a little bit about what we need to do in here. So there's another video on plasmids. But one of the things that will in that video that it highlights is the importance of having a multiple cloning site in your plasmid, which is abbreviated to MCS. Okay, so multiple cloning site is on our plasmid. This is the plasmid. We'll have a region on the plasmid where we have lots of restriction sites, sort of one over the other. So we've got multiple restriction sites. Okay, and you can watch the restriction enzyme video to give you a little bit more about how restriction enzymes work. So we might have these different sites. So let's say we've got BAMH1, we might have then ECHO-R1, we might have ZO1, we might have SP1, we might have Hindi3, and SAL1, let's say. Okay, so we've got these different restriction sites in the plasmid, and they're all pretty close together. Okay, so at the central point of the plasmid, there's all of these different restriction sites at the same place. Okay? And that's a really good thing because that gives us options as to which restriction site we cut open our plasmid with, and that makes our cloning process much more flexible. Okay, so the first thing you need to do is to ask, well, which restriction enzymes am I going to use? Because I've got a choice in there. Okay, so I've got 6 different restriction enzymes that I could potentially use in my reaction. Okay, so in order to think about which enzymes to use, there are a couple of rules. So the first rule is something that will not cut the gene of interest. Okay, so I need to know the sequence of my gene of interest in order to make this decision. So here's my gene of interest. Okay, so that was this is a 2 kilobase system. So I'll do this in pink to keep the colours consistent. Okay, so I've got 2 kilobases of sequence. Okay. And that I've got from my whatever organism I'm interested in, but that will naturally have some restriction sites in it. So every time a restriction site is only 6 base pairs long, so those sequences will come up at random in other genes. Okay, so the first thing we do is to go through and work out what restriction sites are in my gene of interest. Okay, so let's say here I've got ECHO-R5, for example. Here I've got PST1, and here I've got ZO1. Okay, so just naturally in the sequence, and there's online tools that you can do this, you can find out which restriction enzymes are naturally going to cut in your gene of interest. Okay, and we can't use those for our cloning. Okay, so we couldn't use ECHO-RV, we couldn't use PST1, and we can't use ZO1, okay, because if we do those and we digest it, they'll cut my gene of interest up, and then we've not got the whole gene, okay, so we can't use anything that's going to cut inside the gene of interest. So that ZO1 there means that I can't use ZO1 in my multiple cloning site. That's already been excluded for me because it's in my gene of interest, okay. The enzyme should be in the multiple cloning site and nowhere else in the plasmid. Okay, so I'm going to assume for the moment that these restriction sites, this is the only place that they occur in the plasmid at this multiple cloning site. So I'm just going to put that as multiple cloning site there. Okay. If the BAMH1, for example, occurs somewhere else in the plasmid, then we can't use that either. Okay. So at the moment, any of these are good except for ZO1. Okay. Then the next thing that we need to think about is, well, let's think about first how we get the restriction sites onto our gene of interest, because what we want is to have the same restriction sites on the end of our gene of interest. And this is where the PCR step comes in. Okay, so if you did regular PCR, just to see whether a gene was present or not, then what you would do is to design primers for each end of your gene. Okay, so you'd have a forward and a reverse primer. They would copy your gene of interest. You could then you put them through the thermocycler and you extract them on the gel. Okay, so your regular PCR, you just have something that's complementary to your gene of interest. The primer would start at the start of your gene of interest, end at the end of it. and then that will be good enough. What we do with cloning is to mod this a little bit so that we have a cloning primer. And that cloning primer is a little bit different. So it has the same sequence to start with. So let's put that in the blue. So it's got the same bit as the regular PCR sequence if we're trying to amplify the whole gene. But then on the ends of it, we actually make the primer longer than it needs to be. And on that primer, we add a restriction site. So we can then add, for example, let's add a BAMH1 site. Okay, so our cloning primers, so our regular PCR primers usually about 20 base pairs. Cloning primer would be about 30 base pairs and would work like this. Okay, so let's just look at the front end of this gene of interest in a little bit more detail. So I'm kind of zooming in. So we can look at it a sequence specific way. Okay, so let's say at the beginning of the gene, so at the beginning of the gene, we've got ATG, CC8, CC8. There's the start of the gene. Okay. So you might remember that ATG is the translational start site, so that's where the ribosome is going to start. Okay. So that is the gene of our gene of interest sequence. Okay. So what's our primer sequence going to be? Okay, well, the beginning of the primer sequence is going to be exactly this sequence here so that it recognises that gene specifically. So I'll just do there. So we've got ATG, CCA, TCC8T, and so on and so forth. Okay, I'm just showing the first little bit of the gene. Okay, so actually my primer, I'd have about 20 base pairs of gene specific sequence. I would then add some things in artificially. Okay, so I would add, so let's say I want to add the sequence BAMH1, so I want to add a restriction site there. Okay, so the BAMH1 sequence for cutting, so BAMH1 cuts at the sequence, so it's G, G, A, T, C, C, so C, C, T, A, G, G, if I've got both strands. BAMH1 is an example of a restriction enzyme that gives you sticky ends, so it cuts like this. Okay, so what I need to do is to add the BAMH1 sequence to my primer, so that I've introduced that restriction site. Okay, so I'm going to add it on. So I've got C, C, T, A, G, G. Okay, so I've got my restriction site. So I've artificially added that to the beginning. Okay, so this won't hybridize with the gene sequence. It will just sort of flap around a bit, but that's absolutely okay. This bit will bind to the gene of interest. This bit will kind of flap around a bit. But it doesn't work if you just put the restriction site on the end. The restriction enzymes don't cut very well from the very end of the DNA. So what you also usually do is to put in 6 to 10 base pairs. So, you know, let's just say it's A, T, 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 A. Doesn't really matter what it is. Okay, so we also have a 6 to 10 base pair leader sequence. And that's there so that the restriction site isn't at the end. So what the sequence is here doesn't really matter as long as it's 6 to 10 base pairs. We've then got our restriction site, which in this case is BAMH1, okay, and then we've got our gene of interest. And you usually want to make sure that you start with an ATG because that is a start codon there. So that will ensure that your gene of interest can be translated. Okay, so we've got an 6 to 10 base pair lead sequence. We've then got the restriction site, so we've artificially added a bit of DNA at the end, and that is going to be the restriction site, so in this case it's BAMH1, and then we've got the gene sequence including a start codon. Okay, so that would be my sequencing primer, okay, or my cloning primer, sorry. So that's the primer design. Is in there. So this means that I can add any enzyme, any restriction site that I like. I can artificially specify. I can just tell the manufacturer to make a primer with whatever sequence I like. I type in the sequence that I want. I send it off to the company, and they make the primer with that particular sequence. So I can specify whatever sequence I like in there. Okay. So now I can think about my gene of interest. And I can think about what restriction sites I want to add to my gene of interest in order that I can get it into this plasmid. Okay. Can't use ZO1 because that's already been excluded. Okay, we can't do that. Okay. So now what I want to do is to pick enzymes that are in the multiple cloning site. Okay, so let's say we start with BAMH1. Okay, so that's in the plasmid, and it's the start of my gene of interest. When we digest those two things will then be able to stick together. Okay, and then I need to pick another one for the end. Okay, and this leads to something that will be important to think about. Allows for directional cloning. Which I'll talk about in a second. The other thing that you sometimes use, and we don't need to worry about this too much, is to use compatible buffers. So different restriction enzymes like different buffers, and it works well if you pair them of two that like the same buffer. And the biotech companies that you buy these things from have tables of what likes which buffer. So we won't worry about that too much. But what we will have just a little bit of a think about is directional cloning versus random cloning. So random cloning is non-directional. And let's think about what that means. Okay, so we've got random cloning. So if we do random cloning, then what we would do is to cut with BAMH1 in our plasmid. And on my gene of interest, I put BAMH1 at both ends. Okay, so I'd put BAMH1 and BAMH1. Okay, so I could do this. I could choose to put BAMH1 on both ends of it. Okay. When we digest that, then what will happen is the plasmid will open up like that. Okay, so again, we've got the BAM site at each end. Okay. And the gene of interest can go in. We ligate them, we stick them back together. They've got sticky ends. They're both using the right enzyme, so that's okay. But there's two different things that could happen. So this gene of interest has a directionality. The sequence reads from left to right. The polymerase needs to read it in the right direction in order to make something. So if I do random cloning and I've got the same enzyme at both ends, what happens is that could go in that way, or the opposite thing could happen. It could go in the other direction. Okay, so the sequence could flip over, okay, and it could go in either direction, okay, which might be a problem for you in your cloning. So let's imagine you've got a promoter there, so you've got a gene control region that you're wanting to be responsible for the expression of this gene, okay, so that's a promoter, okay. In this way, that'll work. That's the correct way, and that will work. But 50%, just by chance, will be incorrect. The gene has gone in the wrong way. Okay, so random cloning means that 50% of your clones are going to be wrong. okay, which is a bit annoying. So the other thing that you can do is to do directional cloning. Okay, so we've chosen BAM-H1 to go at the start. We're not going to choose BAM-H1 to go on the end because that will have the random cloning effect. What we're going to have is two different enzymes that we pick for our cloning strategy. Okay, so now here's the plasmid. Okay, so we've got the BAM-H1 site, and now let's say we've got let's use SPA1. Okay, so SPA1. Let's say we choose that one. We could choose any of the others actually, except for ZO1. Okay. So we've got SPA1. I'll just put the others in there. And now we've got my gene of interest. I'll put BAM-H1 at the beginning. And I'll put SPE1 at the end. Okay, so just using this primer design process, I can put whatever sequences I like. So I'm going to have BAM-H1 at the beginning, SPE1 at the end. Okay. Now, when I digest that, the plasmid will have BAM-H1 at one end, and it will have SPE1 at the other end. Okay, so now when I put my insert in, so I put those two together and I religate them, it can only go in one direction. You don't get the opposite direction happening. Okay, so because we've used two different enzymes, we chose BAM-H1 and SPE1 in this case. Okay, we use the same on our gene of interest. That means that it's directional. It can't go in backwards because SPE1 won't stick to BAM-H1 and vice versa. BAM-H1 will only stick to BAM-H1, so those are both at the front, and SPE1 is both at the end. So my gene of interest, I'm going to use BAM-H1 and I'm going to add SPE1 as my added restriction site at the end of it. Okay, so I could have used Echo R1 and Hindi3. I could have used Echo R1 and SPE1. I could use BAM-H1 and Hindi3. Doesn't really matter as long as they don't cut your gene of interest, which is why we excluded ZO1. It's in the multiple cloning site and nowhere else in the plasmid. It allows for the directional cloning, so the order that it's in the plasmid is the order that we put it in the gene of interest. So BAM-H1 at the beginning, Echo 1 at the end would work. Echo R1, then BAM-H1, we get it in backwards. Okay, so hopefully we allow for directional cloning. And as I say, just technically, it's a better idea to choose enzymes that have compatible buffers. Okay, so choosing your cloning strategy takes a little bit of brain power. Okay, so you've got to think about what's in my gene of interest, what restriction sites can I exclude because they're already in my gene, which restriction sites are in my multiple cloning site, do I exclude any of them because it's in the gene of interest, and then which ones do I add so I get directional cloning, so I've got my insert is guaranteed to be going in the right way. And to do this, it's PCR. We design our primers, so they start with the gene of we've got the gene of interest, we've got an ATG, so we've got a start codon, we've got the restriction site, and then we've got a leader sequence just to make sure that the whole thing works.